Thanks everyone for coming. We'll start in a couple minutes. Good morning, everyone. Thanks so much for coming to today's A. Watson Armour seminar series. Before we start, I'd like to acknowledge the Field Museum resides within the traditional homelands of the Three Fires Confederacy, the Ojibwe, Odawa, and Potawatomi. The museum recognizes and is grateful for the original peoples who laid the foundation for the city of Chicago and for the diverse indigenous nations that reside in Chicago today. And throughout the museum, as you know, people are committed to building genuine, equitable partnerships with indigenous peoples. Today, it's my great pleasure to welcome to the Field Museum and introduce to all of you today's Armour seminar speaker, Dr. Isabel Loza Rivera. Dr. Loza Rivera is currently a postdoctoral researcher at the Morton Arboretum, where she works in the Global Tree Conservation Program Department. She did her undergraduate work at the Universidad Mayor de San Andrés in Bolivia, and then did both a master's and a PhD in ecology, evolution, and systematics at the University of Missouri, St. Louis, in their program partnership with the Missouri Botanic Garden. Isabel started her scientific career working in one of the most complex plant communities on Earth, Bolivia's Madidi National Park, with 12,000-foot elevational gradient coming down from the high Andes all the way to the Amazonian lowlands. And she was a part of a team that did it all. She did the field work to set up and survey hundreds of plots along that elevational gradient, did the museum work to identify and process tens of thousands of plant specimens that came out of that field work, and then did the analytical work to turn that data set into new understandings of how Andean and Amazonian plant communities are assembled and change with elevation. In her graduate work, Isabel examined these same questions at broader scales, spatial scales, taxonomic scales, time scales throughout Central and South America and throughout the Holocene. And more recently, she's applied these skills in her postdoc at Morton to ensuring that we know enough about plant communities to protect them effectively. So in matters of plant community assembly and plant conservation, Isabel is a true expert, and it's a privilege to welcome you to the museum today.
Hello, everybody. Thank you so much for being here. I'm very happy to be here and excited to show my research to you. And thank you so much for your lovely introduction. And thank you for inviting me to give this talk. As Dr. Pittman said, I'm I'm from Bolivia, and today I'm going to show you my research about woody plants in neotropical forests where being rare is common. So I work in Bolivia for around more than 15 years, collecting data and doing floristic expeditions in the Madidi National Forest. And before I give you my research, I wanna show you the Bolivian tropical forest. These two pictures are from my one of my study sites. And when I started working there, since I was an undergraduate, one of the most surprising things for me was the fact that most species are represented by few individuals in the landscape. As you can see in your picture in your left. You can see there, can you see now? Oh, the pointer is here. So here, for example, this species of palm dominates the landscape. This species is Oniocarpus batawa. And there are other species over there in this landscape that have few individuals. Those are the rare species. So then I was very surprised about how these landscapes were having these very dominant species and many species that are rare in numbers. Then in 2013, this paper came out talking about the hyperdominance in the Amazonian tree flora. And one of the authors of these papers is Dr. Nigel Pittman. In this paper, they use a huge data set of tree plots in order to estimate the population size of the species. So as you can see here in this picture, there is here, this white line over there is the polygon of the study area, and each dot over there is the plots that they use for the estimation of the population density. And the most striking result was that only around 227 species make up 50% of individuals in the Amazon. Here you have a rank abundance diagram of the tree species extrapolated to estimate the size of the tree flora. In the x-axis is the species rank, and in the y-axis is the population size. And one more time, only 227 species have a large population. These species are the hyperdominants. And the rarest species that are around 11,000 species account only for 0.12 of the trees. So most of the species in these ecosystems are rare. So then I became fascinated about why species are rare and how they could escape extinction risk. But why should we care about rare plants? We should care about rare plants because they are more vulnerable to extinction risk, and understanding how they can escape extinction risk can be the foundation for conservation initiatives. In that sense, there are different variables that one can use to define if a species is rare or not. The most common variables are geographic range size, local abundance, and habitat breadth. And each of these measures of rarity are thought to be negatively related to extinction risk, as shown in this figure, where in the x-axis is one of the measures of rarity, and in your y-axis you have extinction risk. I'm going to explain the definitions of these three measures of rarity and also give you the relationships to extinction risk. Geographic range size is the geographic area occupied by a species. Here you have two species. Species A have a wide geographic range represented by that red area, and species B have a narrow geographic range size. Geographic range size or small geographic ranges are particularly likely to be affected by adverse conditions. For example, in this graph, you have two species. One of them is Ardisia pleurobotria over here. And the geographic range size of that species is very restricted to Costa Rica. The other species is Cyathea caracasana that goes all the way down to the Andes and to South America. So if you can imagine if an event of adverse condition can happen here in Costa Rica, so these species will be more vulnerable to extinction risk than this one because this has a wider range. In the same sense, local abundance is the number of species in an area. As we can observe here, where we have two different species, one of them has low local abundance and the other one has high local abundance. Local abundance is also related to extinction risk because small populations are vulnerable to demographic and environmental stochasticity. And in the same way that in the last example, you have here Dendropanax jungacensis that have few individuals, and Socratea exorrhiza that has a lot of individuals. So if something happened with the demography of this species, so if some environmental stochasticity happened over here, so this species would be more vulnerable to extinction risk than this one, just because this has more numbers of individuals. The last one is the habitat rate, that is the number of habitats that species can occupy. In this cartoon, I'm showing you the different species. One of them occupy 3 different environments that are represented here by these 3 different colors. And one of the other species is just in one of the habitats. Habitat rate is also related to extinction risk because species that occupy a small number of habitats are most vulnerable to environmental change, which in this case for Cereus jungacensis over here, that only appears in one habitat, this species would be more vulnerable to extinction risk than Myrcina coriacea that appears in 3 different habitats. So in that sense, one of my research questions was if geographic range size, local abundance, and habitat rate are related with each other. But why we should care about this relationship? We should care because if there is a positive relationship between geographic range size, local abundance, and habitat rate, that would mean that if one species is rare in one variable, it would be rare in the other variables, right? So in that sense, one species that is rare can be 3 times vulnerable to extinction risk just because of this relationship. These relationships were proposed by Brom 1984. However, if there is no relationship in this among these variables, a species can be rare in different forms. So, and it might or could escape extinction risk. And this was proposed by Rabinovich, 1989. She proposed 7 forms of being rare. She did a combination of these 3 variables and classified species into these different categories. She categorized species having large or small geographic range size, wide or narrow habitat specificity, and large or small populations. And she classified each species on these categories. So these are the 7 forms that she describes that a species can be rare. This box over here represent the species that are common. So they have wide habitat specificity, large geographic range size, and have a large population size. For example, in this, we can see that, for example, Rhizophora mangle over here that has large population geographic range size and large population size has narrow habitat specificity. So this species, for example, is rare in one way only, but common in the other two. I researched this question in the tropical forest of Bolivia. And my study area was in the Madidi region that is located in the north part of La Paz over here. And this region is composed by three national parks that are the Pilon Lajas National Park, the Madidi National Park, and the Apolobamba National Park. This region has different ecosystems: lowland forest, mountain forest, dry forest, cloud forest, and polylepis forest. This last ecosystem is found at very high in the mountains, like around 4,000 meters sea level. And the name of this forest is because the polylepis genera is dominant in those ecosystems. That's how we call it, the polylepis forest. So I used the data from the Madidi project. The Madidi project, I used to work there for many years before I came to the US, and it's a collaborative project between the Herbario Nacional de Bolivia and the Missouri Botanical Garden. And this project has been around for more than 20 years, and right now they are in the 22nd year. And we used to go to this region to do floristic inventories. As you can see here, some of our expeditions were challenging. The topography is really challenging over there. Sometimes we kind of walk 3 days just to get to the site, and we camp for a month in order to get all the data that we needed for that region. So each expedition was very challenging, but very rewarding also. So we used to have a lot of mules with us, carrying our equipment, and between 10 and 14 people helping us, and also the researchers and the students with us. So it was a huge expeditions. I did this for almost 10 years of my life going there. So it was really rewarding. So this project established 2 types of tree plots, but for today, I'm going just to talk about the small tree plots. We have in this gradient over here, the gradient of it's between 200 to 4,000 meter sea level. We have around 400 small tree plots that are 20 by 15 meters. And the area is 0.1 hectare. And all the individuals greater than 2.5 centimeters of DBH were measured on those plots. We recorded around 1,000 plant species and also around 100,000 individuals in those plots. And in 10 years or 15 years of expedition, we also got more than 100 new species for science in this region that the researchers over there are still describing for science. So for my research and the question that I have, I measure the 3 variables of rarity. So I measure local abundance for each of these 1,729 species. And local abundance is the mean of the number of individuals across occupied plots. Geographic range size was measured as the species extent of occurrence. And I download from Tropical's database that belongs to the Missouri Botanical Garden all the specimens records, and I just draw a polygon around those, and that was like the extent of occurrence for the geographic range size. Here you have Cyathea leclerc, and that is the distribution that it has. For habitat breadth, I used a vegetation map, and habitat breadth is the number of habitats where a species is found. So we used this vegetation map that was for the region where it was based on bioclimatic, geological, geomorphologic, and edaphic factors. So to recap, we have local abundance, geographic range size, and habitat breadth that we measure in this species. So what we found is that most species in the Madidi region have a small geographic range sizes, as you can see in this histogram. In the x-axis, you have the geographic range size, and the y-axis is the number of species. We also found that many species have low abundance, as you can see in this other histogram, where local abundance is in the x-axis. And also, a lot of species are just found in one habitat or two. So, which is shown here in the last histogram, where the x-axis is habitat breadth. These patterns have been also reported for other regions and other organisms. But coming back to my research question, are geographic range size, local abundance, and habitat breadth correlated with each other? Yes, they are, but in a different way that Brown 1984 suggested. I found a negative correlation between local abundance and geographic range size, as you can observe here. The x-axis is local abundance, and the y-axis is the geographic range size. This relationship was significant, as you can see here. However, I recognize that the Pearson coefficient is low. I also found a positive correlation between habitat breadth here in the x-axis and geographic range size in the y-axis. And this correlation was significant, but as in the other case, the Pearson coefficient was also low. And finally, I also found a positive, significant relationship between habitat breadth here in the x-axis and local abundance here in the y-axis. The main result that I want you to take home is that these results suggest that species with narrow geographic range sizes can compensate for this type of rarity with high local abundance or more uniform distribution of abundance within their range. So it seems that this pattern is also found specifically in high proportion in other assemblage where they have high proportions of narrowly distributed species, as the Madidi region of Bolivia. And this could mean that some species in the Madidi region of Bolivia, even though they have restricted geographic range sizes, they have a lot of numbers of individuals. So maybe they could escape extinction risk in that sense. Okay, what we learned so far is that some plant species can compensate being rare by in one rarity axis by being common in the other one. So in that regard, they can escape extinction risk. But another important question in this context is the extent to which rare species are concentrated in some plant families or groups of closely related species. In other words, is geographic range size, local abundance, and habitat breadth evolutionarily conserved? And these type of studies are of interest because they provide insights into how extinction risk is distributed across a given phylogeny. So to address this question, first I'm going to show you this phylogeny that is a cartoon. of the relationship of these 8 species. And you can see here that the rare species are these red diamonds, and the common species are these blue triangles. So you can see here that the concentration of rare species are in this clade, right? So if these species are most vulnerable to extinction risk and something happened and we lost these species, we will lose this part of the evolutionary history of plant species. So it is very important to know where these species are distributed in the phylogeny, because we can start thinking about conservation initiatives for this specific group of species. But why could closely related species be similar in terms of rarity? Here you have, I will give you one hypothetical example. Here you have in your left a phylogeny with a phenotype that is related to dispersal ability. In this case, how big or small a seed can be. So seeds that are large and heavy will disperse not further away from the tree, right? So that will turn in having narrow geographic range sizes. However, small seeds that are light and tiny can disperse further away. So that will mean maybe that that species could have a bigger geographic range size. So in this case, these clades over here have narrow geographic range sizes. So because so rare species are concentrated in those 2 clades just because geographic range sizes is narrow. So to answer this question, if rare species are concentrated in some group of closely related plant species, we use the same data set from the Madidi region and the same measures of rarity, geographic range size, habitat breadth, and local abundance. Since we needed a phylogeny to answer this question, what we did is to generate a phylogeny at the family level only, because we didn't have a resolved phylogeny to the species level. So we put polytomies of genera and species at the tips of the phylogeny. We generate this phylogeny based following the angiosperms phylogeny group, APG. And we also compiled published estimates of node ages for 74 of the nodes in this phylogeny. Here I'm showing you the phylogeny that we got for the families of the Madidi region over here. And we used different metrics to test if rare species are clustered in certain clades. But for today's presentation, I will only describe the variance partitioning among taxonomic levels. To do this analysis, first I classified each of the species that I had in the data set into these different levels of taxonomic, into these different taxonomic levels. This information came from Tropicals, that is the database from the Missouri Botanical Garden. In the analysis, I have 6 taxonomic levels, as you can observe here, that goes from species to subclass. And in this hypothetical example, the values at the end of the species tips is a rarity axis value or a measure of rarity. This can be, for example, local abundance. This analysis, what it does is it estimates the variation among species within genera, genera within families, and families within orders, and so on. And it gives the percentage of variance in each of the taxonomic levels. Here you have the percentage of variance here. And these dots over here are the observed values. I compare these observed values to a null model. So the null model what it does is to randomize these values over here in order that the evolutionary history of these species are broken, right? And I did that 1,000 times to generate a distribution of values. And for that distribution, we have these box plots over here that represent the null model. So we compare the observed with the null expectation. Here, the variation among species, so this over here, the variation over among species within genera in the observed value is less, the percentage of variance is less than expected by chance. While in these other levels, like the higher taxonomic levels, the variation is more, the observed percentage of variance in the observed value is more than expected by chance, which means that species are more similar among each other than by random. So that means that there are species of rare species that are clustered in some genera and in some plant families. So I'm going to show you now the results of our question if rare species are concentrated in some groups of closely related plant species. So these are our results. So we have local abundance, habitat breadth, and geographic range size. In the x-axis, you have the percentage of variation explained, and you have the taxonomic levels over here. And the same as I explained you in the cartoon before, the dots over here are the observed values. And the box plots over here are the null model. So when you compare the observed value, the percentage of variation explained of the observed value with the null model, you can see that for the species is less than expected by chance. However, for the genera and the family are the opposite, it's more than expected by chance. And this means that there are species, cluster of species that are more similar between each other than expected by chance, which means that rare species are clustered in some genera or plant families. One more time, this is the phylogeny for the Madidi region, or for the plant families in the Madidi region. And you can observe here that rare species are the yellow dots, and common species are the blue dots. The first wheel in this phylogeny is local abundance. The second wheel is habitat breadth, and the third one is geographic range size. And as you can see, there are clusters of rare species across the phylogeny. And this asteroid group, for example, has the most concentration of rare species. So which means that these species, in the case of the Madidi region, are these clades are most vulnerable to extinction risk. So the implications of this study is that due to the temperature increment and habitat loss across the Madidi region, species with low local abundance and narrow habitat breadths or narrow geographic distribution could be more susceptible to extinction risk. And if that is the case, then extinction risk from climate change and habitat loss would be phylogenetic cluster across the species pool of the Madidi trees. And the amount of evolutionary history under threat could be higher than expected by chance. So there is a need for conservation action in these plant clades in the Madidi region. So the two take-home messages that I would like you to take with you is that species with narrow geographic range sizes can compensate for this type of rarity with high local abundance, and that rare species are highly concentrated in a group of closely related species, specifically in the asteroid clade. So the amount of evolutionary history under threat could be higher than expected by chance. And with that, I would like to thank my funding agencies, all the people that helped me in these studies, all the organizations that were involved. And we have more than 30 communities and more than 50 collaborating organizations in this work. This is not only my work, the effort of many. And I would like to say thank you. And if you have any questions, please let me know.
Thank you very much. Are there any questions?
Question. I'm impressed by your discovery and also your studies. I'm from topic, so I'm very much interested about the speciation among local species. And one question that did not mention here. One question, can you hear me? One question nobody put here. I hope you don't feel offended, but there's no mention of epiphyte.
Oh, yeah.
Does that qualify as rare species or dominant species or things like that?
Yeah, that is a great question, and thank you for your question. So in the project, we only had the opportunity to inventory woody plant species. So epiphytic species were not, epiphytes were not sampled. Or,
We'll just pass this around. Hello?
Yeah, thanks. That was super interesting. So I was just wondering about like elevation and like habitat type. Like, did you look at patterns like factoring in like
yep.
different elevations, habitat types? Like, for example, yeah, I'm just curious if this like exponential decline in like abundance and so on. It's like, yeah, do you see that like in cloud forests, for example, or montane forests? You know what I mean, as well as lowlands?
You mean that if I see patterns in terms of abundance in the elevation gradient?
Yeah, is this kind of pattern of like a steep drop off in abundance and range size, is that consistent across different habitats?
Oh, well, I think there are two questions, right? If one species can be dominant in all the elevation gradient, right? There are some species that or some plant families that can be dominated in some, but not in all the elevation gradient. I think I have here like. Oh, sorry, I think I didn't put it there, but yeah, there is a change of abundance for some plant species. Some plant families can be abundant in the lowlands, and then the abundance can change across the elevation gradient. Yeah. Mm-hmm.
I guess I was wondering about the shape of that distribution. Does that shape change across different habitats or elevation?
Oh, okay. You mean this? this shape. I think for most. Oh, okay. You mean that shape?
Yeah,
Yeah.
right.
I haven't tested this with the different elevations as you suggested. That would be interesting to see. But for example, this pattern have been shown in other biomes or ecosystems where species are like very rare. Many species are very rare, and others are very not as. Sorry, I will reframe that. So where species, many species are rare and few species are common. So this pattern was reported for other regions, not only in the Madidi region, but also in other regions in the tropics specifically. But I think what you suggested is interesting to see. I haven't done the analysis by elevation bands, yeah.
I have a question that it's somehow related with this last one, because I don't know in Bolivia, but at least in Brazil, the elevation level in which we are discussing things has some important effect on the level of risk species are under. The lowland level, we have a lot of anthropogenic pressures like farming and urbanization.
Mm-hmm.
So do you have any clue about how this could be related with this extinction risk we discussed here? Because I don't know in Bolivia if the lowland area are under a different kind of pressure, because you said some places are very difficult to access, so they are also somehow protected by this thing and could be just affected by environmental changes or things like this. I don't know.
Yeah, as the same as in Brazil, like there are different pressures in the mountains and in the lowlands, right? In the lowlands, there is more farms, but in the highlands, we have like more mining and deforestation. So all the data that we collected here, where we tried to be in protected areas, but also that they don't have any intervention from anthropogenic pressure. But I will argue like, yes, I mean, there are different pressures there that maybe can be addressed in different ways for the lowland and the highlands. And these kind of studies, I think they are important because they kind of tell you if these species are have this kind of pattern, then you can do more conservation initiatives. For example, when I show like the clustering of the rare species in some clades, so those clades are different distributed in those two different in the highlands and the lowlands, right? Yeah.
Thank you.
Very nice talk. You're probably familiar with the Jansen-Connell hypothesis. Yeah, how would you put that with the patterns that you found here today? Rejected, supported, anything new that could be tested?
So you are asking how can I put the
Yeah, how will you
Jansen-Connell
new data
hypothesis
and will
to
support
my
or
data?
reject that hypothesis?
I am not sure about it. I think I have to think about it. Yeah, I don't have an answer for that. Yeah. Yeah, but it's a good question.
I
Yeah.
think it will probably support it. I mean, I don't know, what do you think, Nigel? Basically, what it says is that the predators, insects and pathogens will attack in a given species, and it will not become dominant in the tropical rainforest.
Mhm.
So that's basically what you're seeing there. But I was just curious.
Just to follow up on the question about conservation, in your talk, you mentioned some implications for conservation.
Mhm.
And now you're doing a postdoc that's focused on conservation. I'm going to take a couple of minutes and tell us a little more about the global tree
Conservation Gone program. Yeah, sure. So I prepared some slides for that. So I'm currently a postdoc at the Global Tree Conservation Program. And this program, what it does is different things. So it does plants like red listing, conservation gap analysis. It does in situ conservation and also ex situ conservation, and does also capacity building. And my work right now is in Mexico and Central America. So I'll be working with oaks conservation efforts in Latin America. And as you know, they are very culturally and economically important, and they are keystone species in many forests. The idea of my position right now is to inform conservation initiatives in Mexico and Central America. For now, what I'm going to do is like to look for areas where there are high endemism and high number of species that are under threat, and try to go to those areas to have more collection of those, and also try to understand if there are rare species, why those rare oak species are going to survive the threats that are there in Central America and in Mexico. Right now, I arrived 6 months ago to Illinois. I'm still trying to find what kind of research question I would like to do in the area. But what I would like to really research is just to have one species that is rare and another that is common, and they are sister species, right? And in oaks, and if I can find that and see which traits makes these common species and these rare species. And in order to that, understand how the rare species can survive over time. Mm-hmm. Mm-hmm. I hope that I answered your question.
Thank you for the talk. My question is about the relationship between the three types of rarity. So you show some graphs and you mentioned how they can compensate with density, but actually seems that the relationship is super, like there's almost no correlation, right? It's very, very small.
Yeah.
So perhaps for any given species, it's not going to be vulnerable in all three fronts, maybe just one or two.
Yeah.
So I was wondering if from your data or from others, other studies, do we know which type of rarity might be more important? What makes them more vulnerable? And I imagine that depends on the scale and the region, but maybe at that scale, like if one of them will make them more vulnerable than the others.
That's an excellent question. And actually, I'm not sure which of the types of rarity would be that. One of the things in my study area is that, for example, local abundance and habitat breadth was measured at local scale, so in the plots, while geographic range size was a regional scale, so for the neotropics. And maybe that would be something that I can look into that. So the scale is important. And also, I think why maybe I don't have a strong relationship is because maybe I have to divide the relationship by families or genera, maybe seeing how the dots the relationship is following the dots. Like, for example, that's what I was thinking, but But to be honest, I'm not sure which of the types of rarity could be more concern for. Yeah, I guess one of the, the one that I would think of is where all the species are rare in all the three, right? That would be more of concern for conservation. But from my results, it seems that really, there are rare species, but there are also rare species that can be rare in one and uncommon in the other. Yeah. Mhm.
Isabel, thank you for a wonderful talk.
Thank you.